Hi, my name is Klocke. I'm a fourth-year PhD student at Oregon Health and Science University, working with Dr. Guanming Wu. This is a brief overview of some ongoing research we're conducting to identify immune cell activity related to T-cell exhaustion in the tumor immune microenvironment. The clinical motivation for this research stems from the early success of a class of cancer immunotherapies known as immune checkpoint inhibitors. While highly effective in a subset of patients, they fail in many other patients, and the mechanisms of this treatment resistance are not fully understood. Research suggests that a process known as T-cell exhaustion, which can be observed in tumor-infiltrating CD8 T cells, may be related to the efficacy of these therapies. To better understand this process, I have constructed a novel bioinformatics pipeline to analyze single-cell RNA sequencing data from human melanoma tumors and from the blood of HIV-infected human patients as a comparison. I first identify CD8 T-cell populations and perform a pseudotemporal ordering of these cells to delineate a trajectory of T-cell exhaustion. Next, I identify gene regulatory network activity in distinct immune cell populations associated with the degree of CD8 T-cell exhaustion in a given sample. Performing this analysis in parallel on tumor and virally infected samples allows us to compare and contrast this biological process between these two disease contexts. At this point, I have constructed the pipeline and it is starting to yield some preliminary results. I have identified some transcription factors in immune cell subsets, including CD8 T cells, B cells, and macrophages, whose activity is associated with the degree of CD8 T cell exhaustion in each sample. As I continue to refine the pipeline, I will be able to better establish the relationship between gene regulation in the tumor immune microenvironment and the CD8 T cell exhaustion process. I will then attempt to validate my findings in independent datasets. Thanks for listening.